Thanks for coming. My name is Josine. I'm a bioinformatician and by now more application specialist at ENPICOM. I'm going to talk to you today about how to organize and visualize a specific genomic data type, which is called repertoire sequencing data, using our IGX platform. Repertoire sequencing is a relatively novel technique, so I was just wondering, maybe up front, how many people are familiar with the technique repertoire sequencing? So the sequencing of specifically B and T cells. Okay, so some of you. So I'll do my best to explain to the rest what kind of data it is and what are the challenges that come with analyzing this specific data type. So a bit about our company. We were founded in 2017. We're located in the south of the Netherlands in a city called Den Bosch. And we focus on the development of software solutions in the field of biomedical sciences, and specifically immunomics, so immune system related big data solutions. We have a strong scientific orientation. Currently, we are about 18 employees, of which the vast majority is either bioinformatician or software engineer. And I think Shai already nicely emphasized that you have to have immunology knowledge and the computational expertise in order to get relevant insight out of these big data sets that are being produced nowadays. Data set sizes are increasing continuously. So we have sort of a unique mix of expertise that is together capable of joining the challenges that we face in the field of immunomics. We were recently lauded as one of the 50 most promising biotech startups of Europe, which to us also was confirming that this is a field that has the potential to grow in the field of immunotherapy. So at our company, we have we develop products, but also we have ongoing projects. I'm going to talk to you today about the IGX Platform, a software solution specifically for the analysis of repertoire sequencing data. But we also do a variety of projects in the field of immunomics, build custom apps for clients with specific biological questions. So these end up as specific analysis apps in the platform that is an off-the-shelf product for the analysis of this data type. So why the immune system? So we all know that immunotherapies have revolutionized the way in which we can treat patients, but we're also aware that there are some major challenges that come with this therapy, such as the fact that it can be very selective, also shown in the data from Shai. Severe side effects have been noticed in patients, and it's very expensive. So from many different aspects, there is interest in precision medicine. So how can we get the right treatment to the right patients at the right time? And in order to give the right immunotherapy to the right patient, we need to have a good readout of what the immune system is like in response to certain treatments, but also to predict before treatment whether a certain treatment is right for a certain patient. And this is what repertoire sequencing is about. So with repertoire sequencing, you look at T and B cells, which are the key players of the adaptive immune system. And the interesting thing about your adaptive immune system is that you have a wide variety of different immune cells. And immune cells bear a specific receptor on the cell surface, which can recognize antigens, which can be virus, bacterial particles, but also altered cells, as seen in cancer. And these receptors that can recognize a wide variety of antigens are the result of a sophisticated process which is called VDJ recombination. And in this process, different gene segments that are encoded in the genome are recombined with the insertion of random nucleotides. And this sort of shuffling process gives rise to a almost 10 to the power 12 possible receptor sequences. And this gives a fundamental challenge to bioinformatics because there's no reference genome that we can use. So what we aim to do with repertoire sequencing is to identify what the immune system in terms of B or T cells is like and how dynamic fluctuations differ in response to immunotherapies. And this has a wide variety of applications in every stage of drug development. So for example, in the more early phases for discovery or drug optimization, High throughput sequencing of these T or B cells can give a wide, rich pool of potential therapeutic targets and a lot of interesting sequence information used to find the optimal drug with the right therapeutic properties. During clinical studies, by measuring specifically the dynamics of the T and B cells, you know better how what effect a certain dose of or a certain drug has on the immune system, which allows you to stratify patient response, patient relapse, and all sorts of clinical aspects that you can measure by looking into the immune system. And also later on in the process, immune repertoire sequencing can give you insight, for example, on certain clones that are present in a certain patient. I think MRD is a clear example where you can really specifically detect a clone that is sort of expect to give a patient relapse after a certain time. So what the general workflow of repertoire sequencing looks like is that you take your sample, select your T cells, isolate the genetic material, and then what you do is that you specifically amplify this hypervariable region. And why specifically this? It's because this variable region is what determines to a large extent the binding to the antigen. So the result, when you sequence this, is millions of receptor sequences that very specifically have this variable region of the receptor in place. And then it's up to the data analysis to make sense out of this data. So to go through these millions of receptor sequences and gain insights from this that will drive the immunotherapy discovery and development. So from a bioinformatics perspective, the challenges that come with this field is, well, of course, the large amount of data. Typically, you get millions of receptor sequences from such an experiment. As I explained, there's no genomic reference for alignment. Random nucleotides are inserted at the junction. Every immune system is unique. So this makes it very difficult for us sometimes to determine, is a difference between two receptors biological diversity, or was it a sequencing error? And because we don't have a reference, that's very difficult for us to determine and requires sophisticated algorithms with immunological knowledge. The immune system is very diverse. You have like a huge amount of different cells in your body, and you only sample a fraction of what's out there. So you have to draw a conclusion on the whole while only looking at a subset. And yeah, last of all, I mean, we encounter a lot of immunological sort of perturbations in everyday life. We continuously are exposed to pathogens. So how do you distinguish the effect of your drug or treatment compared to everyday fluctuations, dynamics that are playing a role in the immune system? So we have immunologists, bioinformatics, and software engineers, and we together have generated the Immunogenomics or IGX Platform, which is a platform, cloud-based, to manage, analyze, and help you to interpret this large-scale B and T cell receptor data. The idea of the platform is that the core is centered around data management, so which means that you have all your receptor sequences in one place, but also tightly associate this to your available metadata. This can be either clinical, but also other experimental data. And once you have all the data organization in place, there are different apps that allow you to do certain types of analysis. For example, IGX Inspect is for quality control. IGX Profile does the actual clone annotation. So it takes in your raw sequencing data and annotates where your clone regions are in the sequence. And IGX Explore facilitates downstream analysis. So visualizing how many different clones you might have in your repertoire. If two repertoires are very alike, so what's the clonal overlap between my patients, and so forth. And the idea is that by combining different apps, you compose your own end-to-end workflow based on the biological question that you aim to answer. And new apps are continuously being developed, and we also custom build apps with clients based on their requirements. So from a management perspective, we noticed that there was a great need for scientists to associate on a very granular level information that they had about the sequences onto the actual data. So they often have metadata and experimental data stored separately. So what we have built is a system in which you can closely associate your metadata to your sequence data at the sample, but also at the clone and also at the receptor level, at the individual receptor level. And this allows you to do a couple of things. First of all, it nicely organizes your data, so you can always find back what you have been working on. Second of all, you can perform very fine-grained searches. So you can look for specific sequences of interest with specific motives or patient features. But what it also allows you to do during downstream analysis is that it allows you to look into different patient cohorts. So you have annotated all your samples with information about clinical phenotypes of your patients. And by annotating this already in your data management system, you can easily stratify patient cohorts in all sorts of different visualizations using the information that you have attached to this in your system. So to come back to sort of the main story, immune repertoires, they contain a wealth of information on ongoing past immune responses. And by specifically looking at the key players of this system, there's a lot of information to be learned that can drive biomarker discovery and facilitate a whole wide range of different applications. And with that, I would like to thank you for your attention, and I would be happy to take any questions. And I would also like to stress that due to the fact that this is novel technology, I would definitely encourage you to think about potential projects where repertoire sequencing could be of added value to discovering trends in response to your immunotherapy development.